Hi, everyone. Welcome to Naturally Immune for Life. Thank you for joining me today. I hope you're all doing well, and I thank you so much for your time. Today, we are going to be covering a landmark study. Very exciting. I wish I had seen it when it came out, but I didn't. But in my recent travels in this topic about SIGA and Tregs and all of that, I ran across it and I was literally astounded. But I have some other news. The book is now published, the new edition, 10 times better. It really wasn't very smart of me to make a full color book last time. Um, it put it made it unaffordable for too many people. But in my mind, I was kind of thinking I wanted it to be kind of like a textbook, part recipe book, part lifestyle book. So, and being a designer, I wanted it to look nice, but it wasn't very practical, and it put it out of reach of far too many people. So I'm very glad this version is much more affordable, um, and you could literally hand it out, and I hope people do hand it out to their relatives, especially new parents, grandparents. This is for all ages, and I'd just like to take a few minutes to talk to you about it, why I included some things, why I left some things out. I'm quite sure it could have been, it's 240 pages. I'm quite sure it could have been 350 pages. No problem. So, what I did leave out is the history of infant formula. Um, I left out what they knew and when they knew it. Um, I also left out a lot of vitamin deficiency uh, syndromes, which are extremely important. Vitamin deficiency syndromes arising from poor microbiota, which is very important for brain health, things like thiamine. So I didn't cover that in the detail that I hope to finish on my site. So anyway, I'm quite sure all of that would have put it at least to 350 pages, if not more. Um, but it's very, very comprehensive. It includes a DIY chapter for a very easy to achieve, easy to afford. Um, Anyone can do it. DIY chapter for a daily lifestyle plan. And that's all I can say about that on YouTube, but it includes that. Um, One thing I was a little hesitant about is how repetitive some things get, as in our microbiome is like another organ, and we have co evolved with these organisms. And it says that quite a lot. And it also says quite a lot to prevent specific conditions. So you know that this is preventable. All these conditions are entirely preventable. But I left them in because they were in every single reference that's included. And I would like people to notice the dates of these references, And you will see that um, maybe at first they just said prevents asthma and allergies. And then as time went on, they would add autoimmune. And then as time went on, they would add cardiovascular. And then as time went on, they would add diabetes type 1 and 2. And then as time went on, they would even add cancers, certain cancers. So I left them in there so that you could see the progression of this research. And I think the oldest reference in there is probably the father of the microbiome, his groundbreaking work where he showed that you could actually transfer starvation from one mouse to another just by a microbiota transfer. So I think that's Dr. Jeffrey Gordon, known as the father of the microbiome, and I think that's probably the oldest research paper covered in the book. And that's why I left all of that in there, even if it seems repetitive, just to show you that it might seem repetitive, but with each progression in this research and with each paper, they began to add more and more conditions, which is what the thesis of GCMAF and Bravo was always, and natural oral immune therapy was all about. And the WHO statement was all about. Um, you should now think of these as T cell mediated diseases, and it includes an entire list, and that even includes things like arthritis and degenerative conditions like arthritis, not just the autoimmune arthritis symptoms, but degenerative conditions as well. So, okay, I'm very happy to have an editor this time who is a much better writer than I am. And she really helped me polish everything. And it's written at a 12th grade level. I tried to make sure of that. And I hope everyone enjoys it. And I certainly hope you'll support me. And most of all, I hope you'll join the cause to help stem this tide of all of these conditions that are entirely preventable. And today in the United States, that includes 90% of 21-year-olds can probably statistically be counted as someone who is going to have IBS, IBD, and then go on to develop other serious conditions leading from dysbiosis. So, okay, they're working outside, so hopefully you don't hear that. But we're going to move on to this week's post, and it is astounding. So, a landmark discovery. HMOs directly regulate human immune cells. And I believe this is from, well, I know it's from Danone research. I'm not sure if it's from their food production or if that's a separate facility. I meant to double check that and I forgot, but it's from Danone. And in the present study, we show for the first time that HMOs modulate human dendritic cells in a microbiota-independent way. I'm sure you hear that truck beeping. Don't want to start over. Sorry about that. In the past decades, there has been a growing appreciation that breast milk beneficially supports the innate immune system and modulates microbiota composition during the first months of life. Breastfeeding is known to reduce the risk of inflammatory diseases, infectious diseases, asthma, and allergic disease, as well as autoimmune diseases. The developing immune system must learn one of its most important lessons: how to distinguish harmless substances from genuine threats. This process is known as immune tolerance. This process, known as immune tolerance, is essential for preventing excessive inflammation, allergies, and autoimmune disease throughout life. For a long time, protection against pathogens was attributed to secretory IgA, which we covered last time, which could capture pathogens from the gastrointestinal system of the child, and later milk components were identified as possible immune modulators. Today, among the various bioactive components in breast milk, human milk oligosaccharides have been suggested to play a key role. HMOs are a diverse mixture of over 1,000 individual oligosaccharides that belong to a different core structures consisting of short-chain and long-chain oligosaccharides, typically present in a 9:1 ratio. In this paper, the term HMOs is used for a complex mixture of HMOs. When individual compounds are referred to, they are labeled with their specific name. Among the most abundant oligosaccharides present in the majority of human milk samples are 2-FL, 3-SL, 6-SL, and LNFP1, with a disaccharide lactose backbone at the reducing end and consisting of glucose, galactose, and N-acetylglucosamine, fucose, and sialic acid. And I want you to note that these are the same substances that are found in infant formula, the same HMOs that are found in infant formula and supplements. So while human milk oligosaccharides are widely recognized for their ability to nourish beneficial bacteria, such as B. infantis, growing evidence shows that these remarkable carbohydrates also communicate directly with the immune system. It is known that HMOs are sampled by the lamina propria residing dendritic cells. In addition, some structures have been detected in systemic circulation, suggesting absorption in the small intestine. These findings indicate possibilities of direct interactions between HMOs and human dendritic cells. A landmark study published in 2019 demonstrated for the first time that HMOs can regulate human immune cells independently of the gut microbiota, revealing an entirely new mechanism by which breastfeeding helps shape lifelong immune health. Dendritic cells act as sentinels of the immune system, and they originate from hematopoietic stem cells in the bone marrow, and migrate through the body's tissues, where they continuously sample their environment for microbes, dietary proteins, and other foreign substances. After capturing these antigens, they travel to nearby lymph nodes and present them to T cells, determining whether the immune system launches an inflammatory response or develops immune tolerance. Regulatory T cells, TREGs, by contrast, are a specialized subset of CD4 T cells that develop primarily in the thymus with additional TREGs generated in peripheral tissues under the guidance of tolerogenic dendritic cells. Rather than presenting antigens, TREGs suppress excessive immune responses and maintain tolerance to harmless substances in the body's own tissues, helping prevent chronic inflammation and autoimmune disease. Until recently, scientists believed that HMOs influenced immunity primarily by feeding beneficial gut microbes, which then produce metabolites that indirectly regulate immune cells, which they do. While this microbiome-mediated pathway remains critically important, researchers wondered whether HMOs might also interact directly with immune cells. To answer this question, investigators isolated a natural mixture of HMOs from pooled human milk and exposed human monocyte-derived dendritic cells, moDCs, to these carbohydrates under carefully controlled laboratory conditions. By removing the influence of intestinal bacteria, they could determine whether HMOs themselves possessed intrinsic immune-regulating properties. The results were striking. As the researchers concluded, in the present study we showed for the first time that HMOs modulate human dendritic cells in a microbiota-independent way. Rather than triggering a strong inflammatory response, HMOs induced what scientists call a semi-mature or tolerogenic state. These dendritic cells remained capable of recognizing potential threats, but they shifted toward producing regulatory signaling molecules instead of inflammatory ones. Specifically, HMOs increased the production of interleukin 10, IL-10, and interleukin 27, IL-27, cytokines well known for promoting immune tolerance, while avoiding excessive production of inflammatory cytokines. Perhaps even more important, these HMO-conditioned dendritic cells encourage the development of regulatory T cells. TREGs. TREGs serve as the immune system's braking mechanism, suppressing unnecessary immune reactions and maintaining tolerance to harmless dietary proteins, beneficial microbes in the body's own tissues. Adequate numbers of TREGs are essential for preventing autoimmune diseases and limiting chronic inflammation. By directly encouraging dendritic cells to generate more TREGs, HMOs appear to participate in educating the developing immune system long before the infant's microbiome has fully matured. The study also demonstrated that HMOs reduced excessive inflammatory responses when dendritic cells were challenged with LPS, bacterial lipopolysaccharides, a potent inflammatory stimulus. Normally, LPS causes dendritic cells to release large amounts of inflammatory cytokines, such as TNF-A, IL-6, and IL-12. However, dendritic cells exposed to HMOs before LPS stimulation produce significantly lower amounts of these inflammatory mediators while maintaining production of regulatory cytokines like IL-10. This suggests that HMOs do not suppress immunity altogether. Instead, they help calibrate immune responses so they remain effective without becoming excessively inflammatory. So, in other words, they promote homeostasis. The researchers also investigated how HMOs communicate with dendritic cells. Their findings suggest that HMOs interact with immune receptors, including toll-like receptor 4, TL4, and DC sign, two receptors involved in recognizing microbial molecules and regulating immune activation. By engaging these receptors, HMOs appear to influence the signaling pathways that determine whether dendritic cells promote inflammation or tolerance. Although additional research is needed to identify which individual HMOs bind specific receptors, these findings provide a biological explanation for how components of breast milk can directly educate the immune system during early life. In this study, human dendritic cells began showing measurable changes in gene expression within 16 hours of HMO exposure, with clear shifts toward a regulatory phenotype and an altered cytokine production observed by 24 hours. These HMO-conditioned dendritic cells subsequently promoted regulatory T cell development over the following several days, demonstrating that HMOs can rapidly influence immune cell programming, albeit under controlled conditions. This discovery significantly expands our understanding of how breastfeeding supports lifelong health. HMOs are no longer viewed solely as prebiotics that feed beneficial bacteria. Instead, they appear to serve a dual purpose. First, they selectively nourish beneficial microbes such as B. infantis, creating a healthy microbial ecosystem. Secondly, they directly influence immune cells, encouraging the development of immune tolerance even before microbial metabolites begin exerting their effects. These complementary mechanisms likely work together during infancy to establish a balanced immune system capable of responding appropriately to infections while avoiding unnecessary inflammation. For adults seeking to restore immune balance after years of microbiome disruption, these findings are especially intriguing. Clinical research has already demonstrated that HMO supplementation can promote beneficial bacterial growth in adults and support the reestablishment of infant-associated microbial functions. Combined with evidence that HMOs also directly regulate dendritic cells, it appears that these unique carbohydrates may influence immune health through two complementary pathways, one by rebuilding the microbiome and another by acting directly on immune cells themselves. Together, these discoveries reinforce the remarkable biological design of human milk and suggest that HMOs remain one of nature's most sophisticated tools for programming immune tolerance across the lifespan. So that's pretty remarkable, right? I'm sorry. It's very cloudy today, so my camera is probably picking up all of the color on the walls. So here is your source. Human milk oligosaccharides promote immune tolerance via direct interactions with human dendritic cells. And then I have for you why similar biological mechanisms are expected, which I cannot cover on YouTube. So you'll just have to come here and read the rest of this post. But I can educate. So what if two natural immune molecules could influence the same immune cells, but in remarkably different ways? And that is human milk oligosaccharides and macrophage activating factors both interact with dendritic cells, the conductors of the immune system. HMOs promote immune tolerance and help prevent excessive inflammation, while GCMAF or MAF supports immune homeostasis by enhancing immune surveillance and the body's ability to respond to abnormal cells. How can two molecules acting on the same cells produce such different but complementary effects? And I want to remind you they both induce homeostasis. How quickly can human milk oligosaccharides begin influencing the immune system? Research suggests they may start interacting with dendritic cells within hours, with changes in regulatory immune signals appearing within about a day. Could regulatory T cells begin developing within days? And how long might it take for these early effects to reshape immune function over weeks or even months? Effects on dendritic cells from both of these substances. Human milk oligosaccharides found in breast milk interact with C-type lectin receptors such as DC sign on dendritic cells. They drive dendritic cells toward a semi-mature state, meaning they induce the secretion of anti-inflammatory and regulatory cytokines, like IL-10 and IL-27, but do not trigger pro-inflammatory responses. This helps dampen allergic reactions and prevents overactive Th1 type inflammation, promoting immune homeostasis for the developing infant. Macrophage activating factors derived from vitamin D binding protein, GCMAF, is an endogenous immune modulating protein. Studies suggest that in addition to activating macrophages, remember you naturally produce vitamin D binding protein in the liver, and it is found in many tissues and fluids throughout your body. Studies suggest that in addition to activating macrophages, it's also found in bovine colostrum, can increase the maturation rate of dendritic cells. It enhances their ability to present antigens and stimulates the release of pro-inflammatory cytokines, driving cell-mediated anti-tumor immune responses. And Th1 in particular is for intracellular responses. So at times that's necessary. So they both work together, but I want to reiterate, according to Say Marai, GCMAF can also be considered inducing immune homeostasis and not an over-inflammatory response, even though it says pro-inflammatory, because it also includes its own breaks. So you don't have to worry about a pro-inflammatory response unless you have a necessary pro-inflammatory response. And as we all know, milk and Hamlet and Bamlet are known for their anti-cancer, anti-tumor abilities. So, okay, I cannot read anymore, but you'll just have to go to the blog post here to read the middle part of this blog post. And I thank you again so much for your time. And the overall message is either way, we're trying to place these HMOs both within the developing immune system and without, as they are now found in infant formula and supplements and therapeutics, and probably soon in some countries, pharmaceuticals. So You have to try to think of everything together. They're they're learning from nature here, and nature obviously is the breastfed infant and human milk oligosaccharides, which are quite one of the most unique and complex substances ever found. But at the same time, they have synthesized bioidentical substances for use outside of that system. So you always have to think of these two things as being together yet apart. And you, even though the supplements will never reach the complexity of breast milk and versatility, because breast milk can modify itself on whether the infant is premature. It modifies itself on whether the infant is male or female, and it can modify itself on an hourly basis depending on the infant's needs. How that happens, they don't quite have all those answers yet, but they know it happens. And obviously, you cannot expect one material substance. Well, we don't know yet because we didn't even know this until this paper, but it would appear you could not expect one substance to do so many different things. But we don't know. We're still learning, and this research is new and it's still developing, and there's still plenty of clinical trials in the pipeline coming up, and I'll be sure to cover them as they do. And again, I would, I would invite you to check out the new book and let me know in the comments what you think. And any and all reviews are appreciated. Um, just let me know what you think, and I'll be I'll be happy to hear from you. So you all have a great day and a great week, and I'll see you next time. Thanks again. Bye.